Does this work? I hope it does. Good. Thank you very much. It's a pleasure to speak to you today. I wish I could have done it in person, but seeing the beautiful landscape there and some people's backgrounds, but this is how it has to be. I will tell you my own personal, very selfish outlook on why you should work reproducibly. My group here in Cambridge, I mean, the one thing we don't do is focus. We work a bit on everything. We work on cancer genomics, on image analysis, and on network understanding of how signal flow in the cell works. And the people who work with me come from all kinds of different backgrounds. There are mathematicians, statisticians, computer scientists, biochemists, medical doctors, physicists. There's all kinds of people. Some of them, most of them work with the computer. Some of them work with pipettes in the wet lab. So what I'm telling you today is what I tell all of them. These are very fundamental principles, regardless of what your own background is and which tools you use in your research. I think this will apply to you too. The key issue I want to talk about is that there should not be any miracles in science. And you might have seen this cartoon before. You see these two people who are, I don't know, mathematicians or theoretical physicists, and they look at this blackboard, and there's a very difficult equation on the top left, and another very difficult equation on the bottom right. And then right in the middle, one of them wrote, then a miracle occurred. And the other one says, well, maybe you should be a bit more explicit here in step 2. And this reminds me of how many papers in my own field look like. If you publish in Nature, Cell, or Science, there are massive data sets, very complex, top-notch, new measurement technologies that people have found out, big data sets. And then you see this 8-page paper in the end in the Glamour journal, and somehow in the middle, what connects them? How do you get from these really complex data sets to the main result of the paper? That should be clear, this should be obvious, there should be no miracles involved here. If I want to read your paper, I want to know how you analyzed your data, how you got to your results. And this will be the key premise of this talk. There shouldn't be miracles. Now, sometimes there are miracles. So let me start with a story of an apparent miracle. It appeared in the New York Times 10 years ago under the title, How Bright Promise in Cancer Testing Fell Apart. You see two people here. They are the good ones. They are biostatisticians from MD Anderson. Any biostatisticians here? Well, you know, biostatisticians, they're usually not the rock stars of science. Honestly, they lead these really minor existences in somebody's basement. They're usually not the ones who get into the New York Times. So why did they do? Well, they wrote a paper in the Annals of Applied Statistics. Again, not a flagship, not one of the flashy Nature type journals, with a very technical title saying, Deriving Chemosensitivity from Cell Lines, Forensic Bioinformatics and Reproducible Research in High Throughput Biology. Sounds all very, very technical. It's actually a fun read, so you should have a look at this paper. With that piece of research, they made it to the New York Times, The Economist, to a US program called 60 Minutes, and many, many other places all over the world. Because what they had found out was a major problem with the research from some very prominent labs in the US. The labs are from the university, from Duke University in North Carolina, and was the group of Joe Nevins. When I started my career here, he was very famous in breast cancer. Nobody has heard from him in 10 years, and this is the reason why. 15 years ago, this group in breast cancer research published in really highly visible publications in the New England Journal of Medicine and in Nature Medicine, some really incredible work. What they said they could do is the following. They take cancer cell lines. These are cells which come from a human, but now they live on plastic, so kind of artificial cell cultures, and they treat them with different drugs. And then based on what genes are active in each of these cell lines, they can predict whether the drug will work in a human patient or not. So you go from a very artificial cell system that you can experimentally manipulate and you draw inference from that that you can then apply to human patients. This was amazing. That's what everybody wanted to do. So this is why they published so well. Everybody got excited, including the bosses, I guess, of our biostatisticians at MD Anderson. And so they went to these two guys, Keith Beggerly and Kevin Coombs, and said, guys, the people in Duke, they have all these exciting things. Can you look into the methods and try to figure out if we can do this with our samples too? So the biostatistician from MD Anderson took the data from Duke and tried to see if they could reproduce the results and if they could also use their approach at home. And they found lots of little problems. So the first one is the following. I told you what the Duke people did is some kind of prediction framework. They want to look at particular lists of genes and say, if these genes are active, then the drug kills the cells. So these genes, they are measured, back then they were measured by something called a microarray, and each gene has an identifier. And that's what you see here in these, I wonder if you can see my mouse, but that's what you see here in these lists. When it says theirs and ours, these are the two lists of particular genes. Theirs is the list that the Duke people found, and ours is the list that the MD Anderson people found by reanalyzing the same data set. There's no overlap between these two lists. But if you look at them for a second, you see a pattern. So Duke reported a gene with an identifier 1881, and MD Anderson found one called 1882. Duke found one called 31321. MD Anderson found one 32322. So there is an off by one error in here. Somehow the lists are pretty much the same, except the index is shifted by one. How did that happen? Well, MD Anderson did the smart thing. They analyzed their data in R. Duke was not that smart. They used the devil's tool, Microsoft Excel. So what they had to do at some point is they had to cut and paste their data, and so to paste and copy it and paste it into some other statistics software. And you see there is a header in Excel which just had the column name on top, and that was pasted like it was a G name, and so just made everything shift. The problem they had is, I mean, the data, the analysis is still valid, but the interpretation of the results completely suffered because you no longer know which particular genes this were and if the analysis made any sense. This is not yet a major issue. That is the kind of problem that happens to everyone when they analyze data 3 times a day. But the Duke data had many, many more problems. So I told you this is a prediction task. So you have some training data, you're in the cell lines, and you predict whether a cell line is sensitive or not. And then you want to go into some patients and see whether the drug is sensitive or not. So you need independent validation data to do that. So what the Duke people had done is they had collected data that other people had published to get these validation data and reported excellent results. The MD Anderson biostatisticians just did basic data analysis, and they plotted the data. And that's what you see here, this box on the left, the heat map. This is just the expression of some interesting genes. Each column is a patient. And you see in the blue and red on top whether the patient was sensitive or resistant to a particular drug. Now, there's some things that you can already spot by eye. One is that not all the columns are different from each other. So you see these highlighted boxes, these long boxes in there. All the columns in each box look almost the same. So somehow this data set doesn't have unique patients in it. There were some things just duplicated. And to make things even worse, if you look into the right pink box at the top, sometimes the same patient was called sensitive and sometimes they were called resistant in the same analysis. The plot on the right is just a simple correlation analysis of the columns in the heat map on the left. And you should see perfect correlation on the main diagonal, but you see all these off-diagonal elements with almost perfect correlation. And that shows you how many duplicated patients were in the data set. This is no longer so easily explained because, I mean, you can just, where did all the patients come from? So if you curate your data set, I mean, you don't just add patients in there and change their labels. This problem with duplicated data and problems with mislabeling patients and cell lines was very widespread in the Duke data set. What you see here in this plot, you see on the y-axis all the different NCI60 cell lines which were used in this study and all the different papers at the bottom that used them. And for each cell line, you see for, I don't know, 6, 7, 8, 9 drugs, whether the cell line was called sensitive or resistant in each of the papers that the Duke people wrote. This should be a very boring plot because a cell line is either resistant or sensitive, and that doesn't change just because you write a new paper about it. But it's not a boring plot. There are all these blocks, sometimes things are red, sometimes things are blue. So it means the Duke people just picked the label they wanted to, which just fit their story in each of these papers. That was a major issue because whether something is resistant or sensitive is important if you then go and apply this to a patient. And by the time all these papers were out, there were already clinical trials in the US trying to do this with patients. So if you then mislabel and switch the label of resistant and sensitive, you might give the drug to exactly the person where it wouldn't work, and that is not good. So after several years, we're now in 2015, there was a lengthy investigation by the US Office of Research Integrity, and it found that the first author of all these studies had engaged in research misconduct, so that person is now officially a fraud. And this was also the end of the last author, because if that happens to your lab, you don't get funding, it's all a bit terrible. So this is what people in my field usually call the Duke disaster. This was the wake-up story in my field, why better reproducibility would help people and what can all go wrong if you produce non-reproducible, sometimes faked results. Now, there's a few things I would like to point out about this story. First, it is a story about fraud, but for me, this is not the main take-home message. When I looked at this, the main take-home message was the intransparency. I trust the people I work with. I don't think they will commit fraud. But the system that had happened in Duke, it was completely intransparent and nobody could see the data except for the first author. Even basic checks like how many patients do we have here? Does that fit to what other people have reported? What labels do we have? Even these basic sanity checks had not been possible to do because nobody had access to the data. So for me, the Duke disaster, while it is a story of fraud, is even more so a story about the problems with being non-transparent in your analyses. This is a major problem because the issues I showed you, you do not have to be a statistics wizard to spot the problem. I mean, the off by one error, everybody could have spotted. The fact that the sample numbers are not right, everybody could have spotted. The data sets they used are so small. Nowadays, you can analyze them on your cell phone. So these were small data sets with simple problems and nobody spotted them because nobody could, because the analysis and the data were not transparent. And the last thing, this is not just the problem of the first and the last authors. I mean, the first author is now a fraud, known as a fraudster. The last author had to early retire, but all these papers had many other authors somewhere in the middle. How do you think that helped them in their career, being somehow connected to this disaster? We had one of those middle authors apply here in Cambridge. We got the CV, we looked at it and we were like, this smells bad. I don't think we take this any further. And that was that, because you're linked to this incredible disaster and you share some of the smell if you want or not. So with this as a background story, lots of people will tell you why you should work reproducibly in your work. And they say, well, it's the right thing to do. Reproducibility is the foundation of science and the Duke people didn't do it. That's why they had to suffer. Being reproducible is honorable. And working openly and reproducibly will make the world a better place. I do not disagree with these reasons, but they sound completely and utterly preachy to me. I mean, I don't know. This is all this idealistic talk about making the world a better place. I mean, look at how I'm dressed. Look at that person is dressed. I don't even wear sandals. So this is just the wrong person. I'm not in my business to make the world a better place. I'm in this business to win it. I want to have a career. I want to have grants. I want to have papers. I want to have money. And I can see it in your eyes. You are a winner too. You're a winner like me. So let me tell you why reproducibility, what it can do for winners. So, my fellow scientists, ask not what you can do for reproducibility, ask what reproducibility can do for you. Well, yeah, my 5 selfish reasons coming up. Number 1, obviously, given the introduction, reproducibility helps you to avoid disaster. If somebody in this Duke lab had checked reproducibility, they would not have become the laughingstock of the cancer field. But even in smaller ways, reproducibility will help you. This is from one of my colleagues in Cambridge. I hope you can read it. It's somebody offering a cash reward because they forgot their backpack in the Penton Arms in Cambridge, in Penton Street. And it's very important for them because it contains, which I assume is the only copy of their research data from 5 years of a PhD. So without the backpack, they cannot graduate. This is quite existential here. But it's not just drunk PhD students in Cambridge. Here is an editorial expression of concern from a few years ago, 2016, about a paper by 2 authors called Anna Lundstedt and Peter Eklöf. What these people had done is they had a study on, for whatever reason, fish eating plastic. Somehow that's important. 2 authors on the paper. And then there were questions about how they had analyzed their data and where they had gotten their data from. And so what the authors did is they notified science, and that's the underlined sentence here, they notified science of the theft of the computer on which the raw data for the paper were stored. These data were not backed up on any other device, nor deposited in an appropriate repository. This in itself is not fraud or anything. It turns out the story is also fraud. So the first author never did the experiment she said she had done. But at this stage, this was not fraud. This is just stupidity. I mean, by having an editorial expression of concern, telling the world that you did not back up your data, you're now, by science, declared a scientific idiot. Don't do that. So in your own research, the problem is, even if you're completely honest, but you did not back up your data, you leave the data, you just lose them in the pub, you have no way to reconstruct how you got to your results, you might just look like these fraudsters. The latest case is just from a couple of days ago. Again, in Science, Fraudulent Data raised questions about superstar honesty researcher. Dan Ariely denies fabricating data, but can't produce records to clear his name. So again, I don't know, jury is out if that person is a fraudster and if there what actually happened there, but they did not somehow keep good track of where their data come from, and so they look a bit like idiots. Now it's super fun calling other people idiots. So let me tell you a story from my own lab. So one of the projects we had a few years ago, we use technologies, or our collaborators use technologies that knock out genes, and they look at the phenotypes of these gene perturbations. And we were trying to find new members of a cell signaling pathway called NF-kappa B. The details don't really matter, but you just go through the genome, you knock out one gene after the other, you see how much they contribute to this particular pathway. This gives you this blue wedge of phenotypes. Some of these effects are very strong. You call these gene hits, and you postulate that they play some role in this particular signaling pathway we're looking at. Problem is, you don't know where in the pathway they work. So what I told my collaboration partners, I said, I can figure this out for you. You have done the first step of finding the hits, but with my computational methods, I can place these hits in a pathway. So we take some known pathway members where we know where they sit in the pathway. We take your new hits, we knock them out. I have a method called NEMS, which puts together a pathway and just connects them to each other. This is what we did, and we got a beautiful figure. In blue, the backbone of this pathway is like the textbook example of how this NF-kappa B pathway should look like. And then all in red, the other things, that's the new hits. And we now have hypotheses on where they should work and who they connect to in the pathway. Excellent. Our collaboration partners went off to the wet lab to do some experiments. and they were very happy. They thought they had actually validated our model, and we wanted to submit to a major journal. While we were writing up the paper, I went to the postdoc working on this, and I said, well, look at the data, get the data into a repository, look at your code, just rerun everything, make sure everything is reproducible. Didn't hear from him for about 2 weeks. After 2 weeks, he comes back and he says, Florian, this is funny. I'm like, okay. He's like, I rerun my code and never get this network again. I get other nice networks and never get this one. Maybe the data changed. I mean, I never noted down which method I really used, or the parameters, or how I did any of this analysis, or how I set my seeds in the analysis, or anything. It's really funny. Maybe we just write a paper about a different network. At that point, this project was dead, because even though we had validations in the wet lab, we had completely lost the story. There was a miracle suddenly happening, and we couldn't explain how we actually came up with this particular hypothesis. There were the data out there, and then there was nothing connecting it to our particular hypothesis, which we had tested. So this paper never got published because there should be no miracles in science. What that taught me is that a publication is more than just a beautiful result. In your publication, you have to go the whole way from your initial analysis all the way to all the different results, all the different figures. Everywhere you back up your conclusions and present your whole beautiful result. You need to do all the steps. And if my postdoc had done this earlier, we wouldn't have wasted years of ours and our collaborators' time in trying to validate something that we couldn't reproduce. So the second lesson is that starting with reproducibility early will help you save time later. Because it's really bad if at the end of the method, at the end of the project, you realize that you have forgotten how you did something 12 months ago. I mean, it's very hard. It's such a time waster trying to figure this out again. So that was my first reason. Sorry, I get all kinds. I should have turned off notifications here. That was my first reason. It's to avoid disaster. The second reason is that reproducibility actually helps you to write papers. So here's another example of my own publications. It's a paper in ovarian cancer in phylogenetics. We were trying to understand the evolutionary process by which a cancer develops. And if you look into the supplementary materials, you see something that we call the SWI file, or now we would call it an R markdown. Doesn't really matter what you call it. This is a file that has text and code mixed together. So what you see there, these gray bars or these gray areas, this is R code. And the other text is just latex text. And so you have the code, but you also have a description of what it does and how you interpret the outcomes. And if you look at the top, there's this button says compile PDF. And if you do that, you get a more or less beautiful PDF, which has your code and the output of your analyses and your description of what you're seeing in a single document. This is very powerful, and you should do it too. The first reason this is powerful is that it's so easy to look up numbers and put them in the paper. You always know that if you change the data in some way, maybe in our case, a new patient would come in and we would include them into the analysis, because everything is in like this one script and nicely annotated. If I rerun this, I will always have the newest p-values, the newest classification results, and the newest data overview tables. So nothing can go wrong with having old versions of the data. Also, it's much more engaging in this way to present your analysis than if you just say, oh, there's a directory with lots of code and figures. I mean, nobody really wants to look at this. But if you go to your supervisor and you say, listen, I've done some analysis, can we talk about this? And it's all in one nice PDF. It's much easier for them to engage with you and have a chat with you about this. And that will make it easier to spot mistakes and avoid disasters again. So with the same collaboration partner, we were looking at, in another study, we were looking at some ovarian cancer data. And so these tumors, they come in different stages. Stage 1 is still okay, and if you're late stage, that is no longer so good for you. That has already metastasized. And some of these survival analyses we were trying to do just didn't work out. Now, because the data were really nicely annotated and described, even us, 2 PIs, middle-aged PIs, could like type in our commands ourselves and analyze the data. And what we found is, what we were expecting were stages 1, 2, 3, and 4. But what we observed in the data that had been given to us by collaboration partners, no stage 1, but stages 2, 3, and 4, a couple of stages called 999, a stage called John, and several stages called XXX. So that means whoever had curated the data had not done a very good job. Everybody could have found this. Our postdocs could have found this. But in this case, we found it because the data was just accessible and easy for us to interpret. And so some really basic problems with the data just stood out to all of us. And that is, again, transparency, some of the things that had been missing in Duke. More than one person can actually look at the data. Now, we have written a paper now, and we submitted one of these reproducible reports. This will now help you, and that's my third argument, this will help you argue with reviewers and help you make reviewers see it your way. So from the paper with the ovarian cancer phylogenetics, one of the reviewers wrote back to us saying, I downloaded the author's data, I tried out a variation of their analysis, and it gave an insignificant result. So it was a question about the robustness of our results. And they had a plot where some line was flat and shouldn't have been flat. Well, we said, thank you very much. There's a reason for this. You didn't look at this particular feature of the data. And if you use this other approach, which is much better suited to the analysis, you see it's no longer flat. There's this bump now, which is what we were interested in. The cool thing here is everybody complains about reviewers and reviewers having some kind of stupid reasons to reject a paper. In this case, we had a very constructive analysis about what is the right way to analyze this data set with us and the reviewers on the same page, all of us talking about the same data set, figuring out what is the best way to analyze this data set. So this was a very engaged reviewer, and we could just argue with them like with normal people. And it was a very constructive, almost enjoyable review process. And that was only because we had actually given them the data and our results in a way that was easy for them to digest and engage with. Now, finally, reproducibility enables continuity. And we've talked about this already in one of the first reasons. Imagine you must have heard something like that. Either you said it yourself or one of your friends said it. My boss said I should continue the project of a previous postdoc, but that postdoc is long gone and left no scripts or data. I mean, what do? I mean, you would just waste a lot of time trying to figure out what the other postdoc did. Wouldn't it be much nicer if that postdoc had left you one of these really nice annotated documents where you could just follow along what they did and where they are in the project? Would be much better. Or maybe you said this yourself. I'm sorry, I did this analysis 6 months ago, which means, well, how can I remember all the different parameters I used in my model? That's what my postdoc told me when we had to kill off this one project on NF-kappa-B. Or maybe you've said this yourself too. I'm so busy, I can't remember all the details of all my projects. Only human, happens to all of us. But again, the solution to this is document what you're doing. The lesson to learn from this is that the first reuser of your data will be your future self, you in 6 months. And the problem is your old self doesn't answer any emails. If you forget to give your future self some hints now, you'll never get them. So write down, document your code, document your analyses, leave your data backed up in a place where you can find them again. So the final reason is that reproducibility helps you to build your reputation. Now look, we're all scientists, we want to have a career here, we need long CVs because the length of your CV, that's the only thing that documents your worth as a scientist, as you know. So how can you make your CV longer? Well, add software and data packages to it. And we have been doing this for a long time. Here's an example from 2012. Bioconductor is like the meeting spot for the more biology, genomics-oriented people working in R. And they have a thing called experiment packages, where we took all our data and all the scripts we used for some paper in Nature Cell Biology and just put it there for other people to reproduce. And we did the same thing again the next year. And nowadays we have this GitHub page. So our latest papers, all the code and the data are on GitHub. If you really want to know how we got to these figures, you can engage with us here, download everything or as much as we can share, which is when there are no privacy issues. Sometimes there are with our types of data, but as much as we can share, all the code is there for you to have a look into it. And this is good because it means when I present my research out there to the world, people trust me because they know if they have an issue with my results, they can just check here. And I get evaluated here every 5 years. There's a running contract I'm on. All of this is always part of my package. This is part of my scientific output that I report, which gives me a longer CV. So if you put all of this together, here are the 5 selfish reasons. You should work reproducibly to avoid disaster because you want it to be easy to write papers. You want your life easy talking to reviewers. You want your own life easy when you continue your own work, and you want to have a good reputation. Are you convinced? Some people are, some people are not. Some people have asked me questions about this. So what? Nice story, Florian, but somebody said, just because you do open science, you're not necessarily right. And that is correct. There is no reason that by being transparent, you're correct. You're just transparent. And there might be many reproducible studies which are completely flawed for other reasons. There might be biases in there that make them very reproducible and still very wrong. But think about it this way. Do you have a dog? Little dog? Lives in your room? If that dog shits in your house, where do you want them to do it? Should it be somewhere where you can easily spot it and clean it up? Or should the dog rub it under the carpet? So if there's some issues with your analysis, you just want to see them early on and easily. And that is the main advantage of open science and transparency. It's not that you're always right. It's just if there's an issue, you can spot it and repair it. Oh, other people said, all this reproducibility stuff is just for nerds with OCD. Well, I am a nerd and I might have OCD, but that's not how I see it, honestly. Because think about the part where you have to document your code for somebody else to read. This is not so much about OCD. This is about putting yourself into somebody else's shoes and imagining what it would mean for them to understand what you are doing. This is much more empathy than it is OCD. This is really putting yourself in somebody else's shoes and thinking about your project as if you were an outsider. So these are much more different social skills you need here. Some people say it's only the result that matters. And yeah, sorry, no. I mean, my story about the NF-kappa B pathway, my own failure in my own lab tells you the opposite, because the result was still there. It's just, it looked like a miracle had happened and we could not publish it. Well, other people say, I would rather do real science than tidy up my data. Yeah, sorry, man, but that you have a very weird understanding of what real science is if working unreproducibly and untransparent and just making up stuff is what you think is real science. I believe that documenting the whole story, how you got to your results, that actually is real science. And if you don't do that, you might not be doing science at all. Well, now it becomes a bit more interesting. People say, of course, I'm on board with what you say, but we can always sort out the code and data after submission. Ooh, that's the devil speaking again. Because the thing is, nobody does it after submission. Once the paper is submitted, people forget about what they have done and they don't care anymore. So if you're a supervisor out there, you just need to crack the whip before submission to make sure everybody has clean projects and cleanly documented code, because there's no leverage afterwards. My field is very competitive and I can't risk wasting time. Well, sunshine, so is mine. I mean, all our fields are very competitive. But the thing is, I don't like wasting time either. And this is why we work very reproducibly, because in the long run, in the short term, I agree, this is an investment of your time and efforts in the short term. But in the long term, if you go for the long game, this will really save you time and sweat and pain in the long term. Now, this might be specific to my field. People say, well, if there's wet lab validation, so people pipetting, who cares if the computer stuff is reproducible? Now, first of all, this somehow assumes there's a hierarchy here where somehow wet lab validation beats my computational predictions, and I do not agree with this. But I feel, to the contrary, it points out how good we actually are in the computational on the computational side in documenting our data and our analyses. If all my friends who do wet lab science were as good in documenting how they did their experiments, if, for example, all lab notebooks would be openly available as supplements in a publication, I think science would be much, much better. So this is not so much pointing out the strength of wet lab validation, it rather points out one of their weaknesses, that they are not yet at the same level of transparency as computational researchers. Excel works just fine. I don't need any fancy R or Python or whatever. Okay, I already called Excel the devil's tool, but I don't want to insult you if you use it. There definitely are uses for Excel. We use it ourselves. It comes usually in very early in projects when we just have to accumulate certain data and we have to take notes on something. But at some point, you have to become professional about your analyses, and you need to minimize the clicking and pasting that you will necessarily have to do in Excel. And for most of my analyses, we run lots of statistical or machine learning models, and we run and rerun them multiple times. This is something you do in a loop, in a programming language, in a scripting language, and you would never do it by fiddling in a program and clicking and pasting and cutting. And so keep your hands scripting, keep your fingers, like use your fingers for scripting, not to click on things in Excel. Oops, where was I? Hello. Mind your own business. I document my data the way I want. Please do. I'm cool with that. That's great. Whatever works for you, do it. I mean, if you write it in Word and just stick it in, that's cool, but just do it. Whew. Finally, the current reward structure does not enough to incentivize data sharing. Yeah, blame the system. Look, I don't know what you really want if you say this. Do you want a pat on the back? Do you want a cookie? Do you want a brownie from me? Because I've given you 5 really good reasons why working transparently is good for you. And you are part of the reward structure. You are part of the system. You will at some point, the older ones amongst you will already review papers, and everyone else will do that soon. You will be able to check if other people did a good job in describing their analyses. And if not, you call them out in the review process and you say there's a major problem with this paper. You can be the incentive you want to see. So if you get me as a reviewer, the first thing I check is there code? Is there some documentation attached? And if not, I say I cannot recommend publishing this paper. So all these incentives are already out there, and you in your own work can help us make them even stronger. So I think we're doing okay on time. So let me talk about the last part here. So all of this was about 5 reasons why you would want to work more reproducibly. But at some point, your paper is out. And maybe you think your work has stopped now and you can just ignore it. It's on your CV, it's another line there, another fancy journal, and you just forget because you're only as good as your next paper. So the standard view of scientific publications is there are a final result. They're pretty static, they're written in stone or, well, baked clay in this particular case here. And few people ever look back at their old papers. The last paper is just less important than the next one. There's an issue with that. And so if you look at some of my own papers, you will find they have corrections. And every time a paper has a correction, look at this. This is one of the flagship papers from my lab from 2012. It has a big red box saying something is wrong with this paper. It's really frightening. I don't really like it having a big warning box appear on the journal webpage saying something is wrong with the paper. If you click on the red box, you will find another 2 sentences explaining what is wrong with the paper. You know what was wrong? We were describing an analysis and we said, we found that survival is better when actually survival was worse. So the data and the results and the plots and everything was okay. It's just in the text, we used the wrong word. And because of a single word, there is a red warning box on the journal page telling people off something's wrong with this paper. Others of my papers are equally flawed. So here's one from a few years later. There's now, well, it's not in red, but now it's in blue. There's a blue box informing readers there's a correction has been published. Something was wrong with the original paper. What was wrong in this case is that actually everything was all right. It was just not in the right order. So figures 4 and 5 had switched in order. We hadn't seen that in the proofs. So again, the analysis, the data, everything is good. It's an incredibly minor error that led to a correction. I find this a very weird way that scientists deal with minor glitches in papers. And it's definitely very different to, for example, what software companies do. So apparently Google pays people lots of money if you report a bug in Chrome, and they will not have a red box appear in Chrome saying, there was a bug found in this code. No, they just repair it, and that's it. Now, everybody makes mistakes. And I think when it comes to the scientific literature, we have to just admit more often that all of us make mistakes. Some of these mistakes might be huge. Maybe there was fraud, who knows? In these major cases, there need to be expression of concerns, corrections, maybe retractions, all of the tools that we already have. But so many more cases are incredibly minor. And so we just need a new way of much more smoothly and transparently maintain and clean up publications without making it look like there was a major issue underlying all of this. So the next stage of reproducibility, at least the way I think about it, currently it says there is a study and there is a publication, and then maybe if you're doing it really well, you have open data and or open code, and other people can try to reproduce your publication. This is already very good. But what I would like to see is that these publications also get improved and updated and maintained over time. And the way I think about this is having something like a new type of submission with an editorial decision and potentially re-review of your paper, where you submit something to a journal and you say, look, we spotted a small mistake in our paper. We have to change a single word. Is it just okay to do this? And in which case, I guess the editor will say, yes, it is. And there is just no red box. It just is an updated version. And this is then shown on the web page. There is maybe an accepted version in 2017. And then there is a minor amendment and maybe another minor amendment. And somebody checked they were really minor and don't really change the story. But currently, I mean, some journals work towards this, like the preprint service. You can have different versions, and some of the journals are actually quite actively trying to have a bit smoother way of improving papers. But all the big journals in my field, they don't do that at all. Like the Science Translation Medicine paper I showed you, big red box. And I think this has to change. People getting more acceptance of the idea that there are bugs out there in the literature and making it much easier to fix them. So with that, let's get to the end. So among the 5, well, at some point I expanded the talk and now I have 7 reasons. And the last one is, if you work reproducibly, you can keep your papers bug-free ideally. So when do you've listened to me now for 45 minutes, when do you need to worry about reproducibility? Well, definitely before you start the project. because there are some skills and tools you have to learn. You need to know how GitHub works. You have to maybe learn all these markdown documents, how to use them, how to write in a markdown language. There's some skills and tools you have to learn. You should definitely do it while you do the analysis and while you still remember how you analyze the data. You should definitely do it when you write the paper, because you want the most the best set of numbers and results, the ones that actually come out of the data, to be in the paper. You should do it when you co-author paper, because I would never trust my co-authors. I mean, I love them, but I would never trust them. So they need to show how they actually got to their results. And I think you should do it after you publish the paper and also when you review other people's papers. So in short, always. Now, how do we get there? Well, to make reproducibility possible, we need everybody. We need PhD students, we need postdocs, and we need PIs, but we need them in different ways. Students and postdocs, you're the people on the ground. You're the people who still program, who still work scientifically, at least in my field. I mean, I have programmed for years. So you need to learn the tools and apply them in daily work. And as a group leader, in my case, my job is to create a culture of reproducibility in my lab, where I tell you, I value the time you put into making it available on GitHub. I value the time that you took to document your code and make it readable. And I just give them the impression, or I really value all the efforts they put into reproducibility. And I think this is the way forward. So with that, thank you very much. These were my 5 or so reasons, and I'm looking forward to your questions and having a discussion with you. Thank you.
Thank you, Florian. Thank you for your very engaging and informative